Hi, students. We will be reading and understanding the sexual reproduction in flowering plants. Let's start with parts of flower. It is modified shoot which is developed from floral primordia. Then reproductive parts of flower are androecium, which is the male reproductive wall, which consists of stamen, and then gynoecium, which is the female reproductive part of flower consisting carpels. Let's have a deeper understanding of how does the structure of megasporangium, stamen, pollen grains look like. So typical stamen consists of two parts, that is filament, which is a slender stalk, if you look here, and then terminal bilobed structure, which is called as anthers. Then a typical angiosperm anther is definitely bilobed. Each lobe have two theca, that's why it's called as dithecous. Each anther contains 4 microsporangia located at the corners, 2 in each lobe. Microsporangia become pollen sacs and are packed with pollen grains. So you have a look of the pollen sacs here. Then the structure of microsporangium. Each microsporangium is surrounded by 4 walls, that is epidermis, endothekium, middle layer, tapetum. The innermost layer, tapetum, which is multinucleated with dense cytoplasm, is used for nourishing the development of pollen grains. Then microsporogenesis, the formation of microspores from pollen mother cells through meiosis is called as microsporogenesis. The sporogenous tissue of microsporangium is differentiated into microspore mother cell or pollen mother cell. Each microspore mother cell undergoes meiosis and gives rise to haploid microspore tetrad. Each microspore mother cell, after having undergone meiosis and giving rise to haploid microspore tetrad, on the dehydration of these microspores, tetrad is dissociated to form 4 microspores. The dissociation is occurred due to dehydration. Each microspore is developed into a new pollen grain. Now the pollen grains. Pollen grains represent the male gametophytes. It is spherical and measures about 25 to 50 micrometer in diameter. It is covered by 2 layers. The hard outer layer is called the exine, is made up of sporopollenin, which is one of the most resistant organic materials which are ever known. The exine has prominent apertures called germ pore, and then the inner wall of pollen grain is called intine. On maturity of the pollen grains, there are 2 cells, one is vegetative and the other one is generative. So 60% of angiosperms pollen grains are shed at this second celled stage, and the others are generative cells which divide mitotically to form 2 male gametes before pollen grains are shed, which is called as third celled stage. Pistil, megasporangium and embryo sac. Pistil is the female reproductive organ, which may be mono, bi, tri or polycarpous, syncarpous, apocarpous. Every pistil has ovary which has 1 or more cavities called as locules, style and stigma. Ovule is a small structure attached to the placenta by means of stalk called funicle. The primordium grows into mass of cells forming nucleus, that is the body of ovule. There are 2 protective coverings of nucleus. Then the basal part of ovule is called chalaza, that lies opposite to the micropyle. The single embryo sac or female gametophyte located in the nucleus, which is developed from megaspore. Let's have a look of the microsporangium. Ovule is a very small structure attached to the placenta of locule called funicle. right here in the bottom. The body of ovule fused with funicle in this region is called hilum, just above that. Hilum is the junction between funicle and ovule. Each ovule has one or two protective envelopes called as integuments. Integuments are covered, they cover the ovule except an opening at the top called micropyle. So micropyle isn't covered, the rest of the ovule is covered with the help of integument. So opposite of micropylar end is the chalaza on the top, representing the basal part of the ovule. In megasporogenesis, the process of formation of megaspores from the megaspore mother cell is called megasporogenesis. In the center of the ovule, there is a mass of tissue called nucleus. Then the cell of nucleus have abundant reserve food materials. One cell of nucleus towards micropylar end differentiated into megaspore mother cell, that is also called as MMC. The MMC undergoes meiotic division resulting 4 haploid megaspores. If you have a look here, the pollination. Pollination is the transfer of pollen grains from anther to stigma. Both male and female gametes are non-motile. Kinds of pollination will be autogamy, geautonogamy, xenogamy. In the first one, pollination occurs with the same flower. There is transfer of pollen grains from anther to stigma in the same flower itself. In geitonogamy, there is a transfer of pollen from anther to stigma of another plant, of another flower of the same plant. So the plant is the same, but there are different flowers which make the transfer happen. In xenogamy, transfer of pollen from anther to stigma of a different plant. So this is the same plant, this is same plant but different flower, and this is different plant. There are several agents which are responsible for pollination. Plant use 2 abiotic agents, that is wind and water, for pollination. These are abiotic factors, wherein biotic would be animals. Majority of the plant use biotic agent for pollination. Few plant use abiotic. So majority is done with the help of bird, insect and bat. Now in double pollination, double fertilization, after entering one of the synergids, the pollen tube releases 2 male gametes into cytoplasm of synergids. The synagamy is one of the male gamete when it is fused with the egg cell to form diploid zygote. Then after that, 2 polar nuclei of the central cell fuse to form diploid secondary nucleus. The secondary male gamete fuses with the secondary nucleus to form a triploid primary endosperm nucleus, which is also called as triple fusion. Since 2 types of fusion, synagamy and triple fusion, take place in the embryo sac, the phenomenon is also called as double fertilization. The central cell after triple fusion becomes primary endosperm cell and develops into endosperm. The zygote developed into an embryo. If you have a look here, this picture depicts the double fertilization. There is a release of sperm cell, then the endosperms take place. This is the third fusion. This is the double fertilization occurring. So what happens after this fertilization? Events of endosperm and embryo development, maturation of ovule into seed and ovary into fruit are collectively termed as post-fertilization events. There is a development of endosperm which takes place before the embryo development. Primary endosperm cell divides repeatedly to form triploid endosperm. Cells are filled with reserve food material and are used for nutrition of the development of embryo. Then it undergoes successive nuclear division to give rise to free nuclei. This is called free nuclear endosperm. Subsequently, cell wall formation takes place and become cellular endosperm. sperm. The coconut water is free nuclear endosperm and the white kernel is cellular endosperm. Now the development of embryo. The development of embryo, the zygote is formed and placed at micropylar end of the embryo sac. The zygote starts its development only after some amount of endosperm is formed. Embryo development takes place in following stages, that is pro-embryo, globular stage, heart-shaped and matured embryo. So then there is maturation of ovule into seed, maturation of ovary into fruit. So the embryo development, the expanding embryo sac, if you have a look here, food from the parent plant, the micropyle, the basal cell transfers to the cotyledon, then cotyledon starts growing and it forms. Zygote divides mitotically and forms proembryo, then it develops into globular and heart-shaped embryo, then horseshoe-shaped, and then matures into one or two cotyledon. The seed, monocotyledon and dicotyledon. Di means two and mono means one. So in monocot embryo, they possess only one cotyledon, that is also called as scutellum, wherein in the dicots is different. In monocotyledon, the part of endosperm retained in mature seed is called as albuminous. The wall of the ovary develops into wall of fruit called pericarp. The fruit develops into the ovary is called true fruit. You see the seed structure of a corn seed here, and these are beans. Thank you. I hope you liked my video. Please follow and don't forget to comment if you understood well.